Okay, let's dive right in. We're talking about organ transplants today, and the numbers are pretty stark. Over 100,000 people just in the U.S. waiting.
It's a huge number. And, the really tragic part is that around 17 people die every single day waiting for an organ that just doesn't come in time.
That kind of urgency.
Yeah.
It really makes you think, what if there was another way? A way to get past the limits of human donation?
Well, that's where this whole field of xenotransplantation comes in, isn't it? Transplanting organs between species.
Specifically, pigs to humans seems to be where the focus is now.
Exactly. And it's because their organs, size-wise, physiology-wise, they're surprisingly similar to ours in many ways.
So this isn't just a backup plan. It could actually be a potential revolution. Maybe even make transplants more predictable, elective almost.
That's the hope. Imagine a reliable supply of organs. It could completely change the game for people needing transplants. No more desperate waiting.
Right. Now, the first question a lot of people might have is, why pigs? I mean, we're genetically closer to primates, aren't we?
That's true, but there are some pretty significant hurdles with non-human primates, or NHPs. Safety is a big one. The risk of viruses jumping species is considered higher.
Ah, okay. And availability too, I suppose.
Yeah, practicality is an issue. Plus, the whole ethical landscape around using primates is, well, understandably very complex.
So pigs offer a kind of better fit in terms of availability, biology, and maybe the ethical considerations seem different to many.
That's how the field has largely approached it. Based on the sources we've looked at, we've got a great scientific overview, an ethical analysis, and a deep dive into the genetic side of things.
Okay, so our mission for this deep dive is to really unpack that. How close are we actually to making pig-to-human transplants work? What have the recent breakthroughs been, and what are the roadblocks still ahead?
And the ethical questions, too, they're crucial. Let's get into it.
So the very first massive hurdle was the body just instantly rejecting the pig organ, right? Hyperacute rejection.
Yeah, HAR. It's incredibly aggressive. We're talking minutes to hours. A standard pig organ just wouldn't stand a chance in a human.
Like the body's alarm bells go off immediately.
Precisely. It sees those pig cells and just attacks. Full force.
And the trigger, according to the research, is these pre-existing antibodies we have. They target a specific sugar molecule on pig cells, alpha-gal.
That's the one. Alpha-galactose 1-GABARY-3-galactose, or GAL for short. Our immune systems are primed to recognize it as foreign.
Why do we even have those antibodies just floating around?
It seems to be from exposure to common bacteria in our gut and the environment. They have similar sugar structures, so our immune system learns to react to them, and unfortunately, that includes reacting to the GAL on pig cells. Cross-reactivity.
Okay, so identifying gal as the main culprit was the first big breakthrough back in the 90s.
Yes, figuring that out was huge. Then scientists started engineering pigs to express human proteins that help regulate the complement system, part of that initial immune attack.
And they also tried removing the antibodies from the person receiving the organ.
Right. Combining those approaches, they managed to push survival in primate models from minutes to, say, days or weeks. A definite step.
Still not long-term, though. What came next?
The next big leap was in the 2000s. Using cloning techniques, they created pigs that completely lacked the enzyme needed to make GAL, the GTKO pig's GAL transferase knockout.
Ah, so taking the target out altogether.
Exactly.
Yeah.
And that made a massive difference. Suddenly, pig organs were surviving for months in non-human primates.
Wow. Okay, so that's a major piece of the puzzle solved.
Yeah.
But immunity is complex, right? It wasn't just gal.
No, definitely not. So alongside the genetic changes, researchers were getting much better at suppressing the recipient's immune system generally. Things like targeting specific T cells and B cells, using drugs like mycophenolate mofetil, blocking key immune pathways like CD40, CD154.
A multi-pronged attack on rejection.
And they kept refining the pig genetics, too. They identified other non-gal targets for our antibodies, things like NuG and SDA antigens, and started knocking those out as well.
It sounds like they're systematically disarming the immune response piece by piece.
Pretty much. And then they started adding human genes into the pigs.
Adding things in, like...
Things to help control that complement system even better. Genes to manage blood clotting, which can be a big issue. Also, things to inhibit immune cells, like macrophages. That's where adding human CD47 comes in. It basically tells macrophages, don't eat me.
A friendly signal.
Sort of, yeah. And anti-inflammatory genes, too. It's become this really complex multi-gene modification approach.
And the results in primates have gotten pretty impressive with all this.
Very. We're seeing reports of pig kidneys lasting over a year in monkeys, life-supporting pig hearts going for 9 months.
9 months for a heart, that's significant.
Absolutely. They even tackled the issue of the pig heart potentially growing too big after transplant by knocking out the growth hormone receptor gene in the donor pig.
Okay, so the progress in beating that immediate HAR and extending survival, at least in primates, seems really substantial.
It really does. It provides a strong basis for thinking this could eventually work in humans.
But HAR isn't the only type of rejection, is it? Even if you get past that initial phase, the immune system can still cause problems later on.
That's right. Once you get past the hyperacute stage, you face other immunological hurdles. There's acute cellular rejection,
Okay, when does that kick in?
usually days to weeks after the transplant. This involves a whole different cast of immune cells, T cells, B cells, macrophages, natural killer cells, all getting involved in attacking the foreign tissue.
So a slower, more targeted attack.
Kind of, yeah. Each cell type plays a role, and then there's also acute vascular rejection.
Vascular.
Oh,
Affecting the blood vessels.
exactly. This tends to happen a bit earlier, maybe 2 to 3 days post-transplant. It seems to involve leftover antibodies, those macrophages again, and platelets interacting.
Leading to?
Inflammation and blood clots forming within the tiny blood vessels of the transplanted organ. It can choke off the blood supply.
Not good. So what are the strategies there?
Researchers are working on ways to calm down the endothelial cells lining those blood vessels and the macrophages too, using things like HLA-G, HLA-E, boosting that CD47 don't eat me signal.
More genetic engineering.
Yeah, often, yes. Or trying to induce the pig organ to produce anticoagulant factors, or dampening down inflammatory signals using things like the A20 protein to block NFKB. It gets pretty technical.
It sounds incredibly complex.
Yeah.
And then, even further down the line, chronic rejection.
Yes, that's the long-term worry, months, even years later. It seems to be driven more by the antibody side of the immune system, the humoral response.
And the solution for that.
Well, that's still being worked out. One idea is using gene transfer to deliver anti-inflammatory molecules like TFPI, CD39, or HHO1 directly into the graft, trying to create a more tolerant environment long-term.
So, multiple layers of immune response to overcome. It's not just that first few minutes.
Not at all. It's an ongoing challenge at different time points.
Okay, shifting gears a bit. Beyond rejection, one of the really big concerns, especially thinking back to why we moved away from primates, is disease transmission, right? Zoonosis.
Absolutely critical. How do we make sure transplanting a pig organ doesn't accidentally introduce a dangerous new pig virus into the human population? That was a major factor in avoiding NHPs.
And pigs aren't virus-free. The sources talk about PRVs, porcine endogenous retroviruses.
That's right. These are tricky because they're actually part of the pig's own DNA integrated into the genome.
So you can't just raise the pigs in a super clean environment to get rid of them.
Nope. Standard pathogen-free techniques don't eliminate PRVs because they're inherited, and they can potentially replicate.
Has there been evidence of these PRVs actually infecting human cells or transplant recipients?
So far, in the studies done in primates and the initial human trials, there hasn't been direct evidence of PRV infection causing disease. But, and it's a big but, the sources are very clear that the risk can't be ignored.
Because the consequences could be catastrophic if one did adapt.
Exactly. The comparison that often comes up is HIV/AIDS, a virus-jumping species with devastating results. Nobody wants to risk repeating that.
That's a really sobering parallel. But wasn't there a massive breakthrough on the PRV front using CRISPR?
Yes, this is one of the most exciting recent developments. Using CRISPR-Cas9 gene editing, scientists have actually managed to go into pig cell lines and disable all the copies of the PRV genes.
All of them, wow.
Yeah, and then they were able to clone pigs from those cells, creating PRV-inactivated pigs. It's a huge step towards mitigating that specific zoonotic risk.
That sounds like a game changer for safety.
It potentially is, for PRVs at least. But you still have to remember, transplant patients are on immunosuppressants.
Right, which makes them more vulnerable to infections generally.
Exactly. So even beyond PRVs, meticulous screening of donor pigs for all known pathogens is absolutely essential. And the type of organ matters too.
How so?
Well, lungs, for instance. They're connected to the outside environment via airways, so there might be a slightly higher theoretical risk there, maybe even for generating new recombinant viruses. Like with influenza, it needs very careful monitoring.
Okay, so the biology is incredibly complex. But what about the person receiving the organ, the human side of this?
Yeah.
Having an animal organ inside you, that must bring up some psychological stuff.
Oh, definitely. And the sources really dig into this, even with human-to-human transplants, allotransplants. Recipients sometimes struggle psychologically, integrating this foreign part into their body, their sense of self.
And the idea is that could be even more intense with a xenotransplant, an organ from a different species entirely.
It seems plausible. They mention the case of Clint Hallam, the hand transplant recipient who ultimately had it removed partly due to psychological struggles. And facial transplant patients sometimes grapple with identity questions.
And maybe the specific organ matters too. A heart, for example, carries so much symbolic weight for people.
Right. The heart is often seen as the seat of emotions, of identity itself. Accepting a pig heart might be a bigger psychological leap for some than, say, a kidney.
That makes sense. And then there's the social aspect. Could people face stigma?
That's a real concern highlighted in the ethical analysis. Could xenotransplant recipients face discrimination or prejudice, being seen as different or somehow less human?
Like the stigma people with HIV/AIDS have faced.
That comparison is made, yes. It underscores how serious the social integration challenges could be. We need to think about supporting recipients not just medically, but psychologically and socially too.
And what about informed consent? Giving someone all the information they need to agree to this kind of procedure must be incredibly complex.
It really is. Patients need crystal clear information about all the risks, including the zoonotic risks, which are still not 100% quantified, and the potential benefits.
And it's not just the individual, is it? Because a new zoonotic disease could spread.
That's the really tricky part ethically. If there's a risk, however small, to public health, does society have a say? How do you balance individual consent and potential collective risk? It raises tough questions about privacy, monitoring, and individual liberty versus public safety.
Definitely a lot to grapple with there.
Yeah.
So with all these challenges, where are we with actual human trials? You mentioned some early steps in 2021.
Yes, 2021 saw some really important first steps, although not in living patients initially. An NYU group took genetically modified pig kidneys, gall-deleted ones, and connected them to brain-dead human recipients.
Okay, what do they find?
They ran the experiment for about 54 hours. In 2 cases, the kidneys actually started producing urine, functioning. And importantly, no hyperacute rejection, no detectable PERV transmission.
There was another group, UAB.
Right, University of Alabama at Birmingham. They did something similar, transplanting modified pig kidneys into a decedent model, someone whose own kidneys were removed, for about 74 hours.
Same results.
Pretty similar. No immediate antibody-mediated rejection, no major clotting problems, no hyperactive immune response, and again, no PRVs detected.
So encouraging signs, but very short term and not in living circulating systems long term. What could they really learn from that?
Well, it was crucial first data. It showed that these modified organs weren't immediately destroyed, that they could perform basic functions, and it gave some initial clues about managing the immune response in a human context. But yeah, limited duration, ethical limits on how long you can go into sedans, lots still unknown about long-term function.
Then came the big one, January 2022. The University of Maryland team transplanted a pig heart into a living patient. That got huge attention.
It really did. A patient with terminal heart failure received a 10-gene modified pig heart.
And he lived for 7 weeks, was it?
Yes, about 7 weeks. Now, that's shorter than some of the long primate survivals, but it was still a landmark moment.
Why was it so significant, even with the shorter time frame?
Because it was the first time a genetically modified pig heart actually sustained human life. It proved the concept was possible, that the immediate rejection could be overcome in a living person. It showed that genetic engineering was having the intended effect, at least initially.
Did they figure out why the heart eventually failed?
There's evidence suggesting a porcine virus, a type of cytomegalovirus, CMV, specific to pigs, might have played a role. It wasn't necessarily rejection in the classic sense.
So potentially something that could be screened for and prevented in the future.
That's the hope. Better screening of donor pigs, maybe even antiviral treatments, but it definitely highlighted that unexpected complications can arise.
So even with these milestones, lots of questions remain. What are the big ones researchers are still trying to nail down?
Oh, plenty. What's the optimal set of genetic modifications? Do we need all 10 genes used in the Maryland case or fewer? Could some modifications even cause problems long term?
Like the human CD47, is that definitely needed?
That's still being debated for solid organs, yeah. And who should get these organs first? Should trials focus only on patients with absolutely no other option?
And getting regulatory approval, setting up safe facilities?
Exactly. It requires tight collaboration between scientists, companies, and regulatory bodies like the FDA. And you need specialized biosecure facilities for raising these donor pigs.
But the primate data is strong enough to justify these early human steps.
The consensus seems to be yes. The success in NHPs with life-sustaining organs provides a solid rationale for carefully moving forward with preliminary clinical studies. But it hinges on rigorous preclinical work continuing alongside and critically on the volunteers who participate and the families who consent in decedent studies. Their contribution is immense.
Constant monitoring, constant research needed.
Absolutely. Safety has to remain the top priority.
Now, there was one other futuristic approach mentioned in the sources: growing human organs inside animals. Interspecies chimeras. That sounds wild.
It does sound like science fiction, doesn't it? But it's a real area of research. The idea is to create an organism, a chimera, that contains cells from two different species.
And the goal is to grow a human organ inside, say, a pig.
That's the concept. Using the pig as a kind of living bioreactor to grow a replacement human organ.
How would that even work? They mentioned blastocyst complementation.
Right. So you take pluripotent stem cells, human ones in this case, that can become any cell type, and you inject them into a very early pig embryo, a blastocyst.
But not just any pig embryo.
No, this pig embryo would be genetically engineered, so it can't develop a specific organ, let's say a pancreas. It has a vacant developmental niche.
Ah, so the human stem cells then fill that gap.
That's the idea. They'd hopefully colonize that niche and develop into a fully functional human pancreas within the growing pig.
Has this actually worked in animals?
Yes, quite remarkably in some rodent models. They've grown rat pancreases in mice and mouse pancreases in rats.
And those organs were functional.
Yes, in the mouse pancreas in rat example, they transplanted the resulting pancreas into diabetic mice, and it actually cured their diabetes, normalized blood sugar. And because the pancreas was derived from mouse cells, no immunosuppression was needed for that transplant.
Wow, that's incredible. Have they done it with other organs or larger animals?
They've had success generating kidneys, thymus tissue, blood cells, even lungs in some animal combinations using this method. And relevant to our discussion, they've managed to grow a basic pancreas structure from external cells in pigs that were engineered to lack their own. So the potential in pigs is there.
But it sounds like there must be huge challenges doing this between humans and pigs.
Oh, absolutely. Major hurdles remain. There's often high mortality in these chimeric embryos. There are fundamental biological barriers between distantly related species like humans and pigs that we don't fully understand.
And just the complexity of organ development.
Right, making sure the organ develops correctly, connects properly. Also, the host animal can limit the size of the organ grown. And human stem cells just behave differently. They're harder to integrate into host embryos compared to rodent cells.
So are we anywhere close with human-pig chimeras?
We're seeing progress. Things like generating human-monkey chimeric embryos show steps towards bridging evolutionary distance. And there have been reports of detecting human liver cells in human-pig chimeric embryos.
But not whole organs yet.
No, not yet. Growing a complete, complex human organ like a kidney or heart inside a pig via chimerism, that's still a very long way off for clinical use. Fascinating science, but definitely future tense.
Well, this has been a really comprehensive deep dive. It's kind of mind-blowing how far the science has come, especially with the genetic engineering of pigs to tackle rejection and the CRISPR work on PERVs.
It really is remarkable progress, from HR being an absolute roadblock just a couple of decades ago to where we are now.
And those first human transplants, even the short-term ones, feel like such pivotal moments. Learning that a pig heart could actually support a human, even for a limited time.
Definitely encouraging, yes. Vital knowledge gained.
But we absolutely have to keep the remaining challenges in focus. Optimizing the gene edits, ensuring long-term graft survival without rejection or other complications, figuring out the psychological and social angles.
And guaranteeing safety, especially regarding zoonosis, that remains paramount.
Plus the chimera route, fascinating, but clearly not a near-term solution.
Still very much in the basic research phase.
So, wrapping this up, given everything we've discussed, the immense need for organs, the incredible scientific strides, but also the remaining risks and ethical complexities, the final thought we want to leave you, the listener, with is this. As xenotransplantation potentially moves closer to being a clinical reality, what ethical and societal considerations do you think should be given the highest priority?
It's a question with huge implications for the future of medicine and something society will really need to grapple with. Lots to think about.